Continue on enzyme kinetic. Okay, enzyme kinetic after this membrane remodeling. Okay, enzyme kinetic. So temperature can affect the protein structure and function. So how? So protein itself has optimum temperature, right, to cause the action. Enzyme has optimum temperature. When achieve the optimum temperature, it will give optimum reaction, and the substrate, the product after the enzymatic reaction occur will be produced. Okay, so but if exceed high temperature, the protein can be denatured, right? So what happened during the denaturation of protein or enzyme? So a lot of bonds, okay, changes in weak bonds, ionization state of critical amino acid. So if you look back into amino acid, we have R group of each amino acid. That R group of amino acid can be altered when high temperature, maybe ionization of that amino acids. Okay, eventually alter the whole protein structure, that eventually that protein or enzyme cannot be used anymore, denatured. Okay, I think you understand this concept. So what happened when enzyme or protein in the cell in the body denature? So there is system, or there is regulation system in the cell in the body. We call it heat shock response. Heat shock response. So when protein denature at high temperature, faster at high temperature, so this accumulation of denatured protein can kill the cell. So too many denatured proteins can kill the cell, can cause damaging effect to the cell. Okay, what happened in the cell? We have these protein molecule chaperones. Okay, we call it heat shock proteins, HSP. So it's a molecular chaperones that catalyze protein folding after translation and help refold back the denatured protein. Okay. Increase in the level of HSP in response to extreme temperature. Okay, what happened is when the temperature is high, okay, a lot of protein denatured. Okay, so that's when this HSP protein will react. Okay, the protein denatured accumulate. It will make this HSF, heat shock factor monomers, in the cytoplasm to trimers, or to bind together, make trimers. Okay, and then they will phosphorylated. This is the process what happened to this protein here, these monomers here. Okay, eventually they bind together, make trimers, and then phosphorylated, and then they will travel to the nucleus, and when arrive at nucleus, they will bind to this heat shock protein. We call it heat shock protein, and transcription happen. It will produce HSP lah. Okay, it will produce the HSP protein. This HSP protein will travel to cytoplasm and will bind HSP. Okay, will bind to that denatured protein and then refold back. Okay, refold back the denatured protein, and then that protein can be used again. Okay, this is the process in diagram form. What happened when temperature is high? Okay, when temperature is high, physical or chemical stress, the protein denatured. Okay, accumulation of this can cause damaging effect to the cell. Okay, when protein denature presence, this HSF protein, HSF monomer, heat shock factor, this is like a stimulus. Okay, this monomer will start to make trimers. Okay, and then once make trimers, they phosphorylated, and then travel to the inside the nucleus here, bind to the gene. What gene? Which gene? HSP protein gene. Okay. Transcription happen to make a lot of HSP protein. When HSP protein produce, this HSP protein travel to cytoplasm, and it will go to the denatured protein and refold or make you correct back the denatured protein. So eventually that protein can be used again. Okay, that protein can be used again. Okay, so the membrane remodeling and also the heat shock response is a tissue remodeling. Okay, tissue remodeling. And the term can remodel tissue is responsible to long-term changes. See, long-term changes in two ways. Quantitative, in terms of number. Okay, more of the same machinery. Low temperature increase number of mitochondria. Okay, this one is the brown adipose tissue. Okay, or apply to the number of fatty acid change. Higher number of unsaturated fatty acid change. Okay, the king's one. Fatty acid change that have double bond will make that tail of phospholipid, phospholipid like layer, phospholipid have bent leg. Okay, in terms of quantitative. Another one is in terms of qualitative. Qualitative, different, alter the type of machinery. Okay, this one example, different myosin isoform in winter. Myosin isoform is in the muscle. Okay, or we can apply to membrane remodeling just now. Two types of phospholipid. Okay, two types. Here, two types of phospholipid. Phosphatidyl chloride or phosphatidyl ethanol anion. Okay, so this one in terms of qualitative regulation. Quantitative regulation, the higher the number of the fatty acid change that have double bond. Okay, so tissue remodeling in terms of thermal regulation is two strategies: quantitative strategies and qualitative strategies. Okay, so this one, strategies for surviving in cold temperature. Mostly the cell membrane remodeling, the heat shock response is surviving or the high temperature. Okay, strategies for surviving hot temperature at cellular level. Okay, or at tissue level. This one, strategies for surviving freezing temperature at cellular level. Okay, so there are two mechanism or two approach, whether to survive in cold temperature, whether to tolerate with the cold temperature. Okay, tolerate, or to avoid the cold temperature. So freeze tolerance, allow the tissue to freeze, encourage ice to form in the body. So what happen is the cell in the body will form ice or ice crystal formation here. This ice crystal form, it will trigger by a cluster of water become freeze, become ice, and then this ice crystal eventually act as nucleator. Once in the cell we have one nucleator, or one ice crystal formed, it will attract other water molecule to freeze also. Okay, so inside cell will freeze. Okay, so So just now the ice crystal, the nucleator that formed, okay, the ice crystal that formed and act as nucleator. So that nucleator will it act on outside of the cell. Okay, produce nucleator outside of the cell to control the location and kinetic of ice crystal growth. Extra fluid freezes while intracellular space remain liquid. Okay, and then another mechanism, the intracellular solute produced inside the cell to counter the movement of water. Once the cell, and then we have the cell, so the outside here start to freeze. So in terms of particle concentration, or the movement of water, the water tend to goes out, right? But to balance back, to encounter this, inside cell will produce intracellular solute. Okay, it will produce intracellular solute inside. Okay, so in that way, suddenly particles or solute inside the cell is higher, so the water can be maintained inside the cell. So this cell will survive in this freezing temperature, despite of outside being crystal ice. For freeze avoidance, okay, solute in general depress the freezing point of a fluid. Okay, that's this one. You can apply just now. The solute inside the cell will cause the movement of water. Will cause no more movement of water outside the cell when freeze tolerance. But this one, solute in general depress the freezing point of a liquid. So there are antifreeze macromolecules. Antifreeze macromolecules, proteins or glycoprotein. Example here, it will depress the freezing point. Due to the presence of these glycoprotein or anti-freezing macromolecules, it will cause the freezing point inside the cell or outside the cell, or the tissue will be much more lower. Therefore, these cells here or this tissue will not freeze when in extreme temperature due to these antifreeze macromolecules. Okay, another one, this freeze avoidant, these antifreeze macromolecules will disrupt the ice crystal formation by binding to small ice crystal to prevent their growth. So what happened in this strategy is, we have cell, okay, suddenly ice crystal form, okay, ice crystal form, and this cell will produce have antifreeze molecules. We make it blue color. Okay, so this antifreeze molecule will disrupt this ice crystal formation, no more ice crystal formed. Okay, at the same time, due to these antifreeze macromolecules, it will depress. The freezing point is 0, for example. So with the presence of these antifreeze molecules, the freezing point become -20. So this cell can survive more in this freezing temperature, okay, due to this solute antifreeze macromolecule.